The overall goal of this procedure is to determine the functional lateralization of language in the brain using a child-friendly procedure. This is accomplished by first applying ultrasound probes to the head to monitor the rate of blood flow in the middle cerebral artery to each brain hemisphere. The second step of the procedure is to monitor blood flow while the participant performs a language task. The third step of the procedure is to extract the average Doppler signal from the left and right hemispheres over a period of interest in which the language task was performed. The final step of the procedure is to examine the difference in left and right activations to extract a laterality index. Positive values indicate left lateralization and negative values indicate right lateralization. Ultimately, results can be obtained that can be used to compare brain lateralization in children at different ages or for groups with and without developmental disorders.
The main advantage of this technique over existing methods such as functional magnetic resonance imaging is that the participant does not have to keep still, and the method is relatively inexpensive and portable. Demonstrating the procedure will be Georgina Holt and Nick Badcock, a research assistant and postdoc from my lab.
This video will demonstrate the use of functional transcranial Doppler ultrasound, fTCD for short, to assess language lateralization in children. The system consists of a multi-dop with ultrasound probes attached to a headset, a stimulus computer that will be used to show videos to the subject, and a voice recorder for auditory responses.